Yeah, so hello everybody to our second virtual action club. And the nice thing we learned is that while we're only there as two-dimensional postage stamps, it does make it a little easier to attend for people further away. And we even have one person, Hide from Japan, joining us, which is very nice. So before I want to introduce Aaron, I just want to repeat again some of my recommendations to make our action club hopefully as enjoyable for everybody and hopefully also as spontaneous with interactions and questions. And the one thing is I would ask everybody who feels comfortable to turn the video on, including your cat, Aaron. So it is just so much more immediate to see faces. And for the speaker, I must say, to see people's faces just makes all the difference in these virtual presentations. The other thing is that Aaron does have a second computer, which is the cat's computer. Where he has all of your faces on the side, so he can see, you know, how he comes across with his cool messages. And also, this gets to the next point, to how do we do our interactions as spontaneously as possible. You either use the hand, raise hand, image, then I can see it and I can then give you the word to ask your questions. Or you also, Aaron sees the raised hand and he immediately can turn to the person who wants to have a comment or ask questions. Please don't use the chat, because in extreme cases, I have experienced that the chat becomes like a parallel line of communication, which I find very distracting because I'm interested in it, but primarily talking of myself, interested in the speaker and would like to hear everybody's comments and reactions together in the plenum. So with that as a sort of recommendation and preamble, let me continue to introduce Aaron briefly, because I think we know him. And in fact, Aaron did give a talk already at Action Club 5 years ago, and it was totally cool, fascinating. So I thought, let's just have him again, because he's been busy the last 5 years and it will not be the same material, of course. So very few words about Aaron. Aaron is currently professor of bioengineering at the University of Pittsburgh. And his previous location was Caltech, where he did his PhD. And he did that, so I just learned, in the Center for Computation in Neural Systems, meaning in CNS. So he worked right in the CNS, the central nervous system, with Richard Anderson, who is, for anybody who works in this field, a very big name. And prior to that, he also did a bachelor's degree, of course. And interestingly, that bachelor's degree was in computer science and philosophy. So I think, and when I heard Aaron for the first time, I always thought somehow he didn't sound quite like sort of the 100% neuroscientist. There was something interesting extra in it, and I attribute it to the philosophy degree that he went through. So with that, Aaron, it's all yours.
Awesome. Thank you, Dagmar. Thanks for having me back. And it is true that there are new things. They're at the end, the way these talks unfortunately go. So I hope that we can go together through some background material that I hope you'll find engaging and informative. And I want to pause a lot for conversation as I go. And at the end, I'll cover some new things. And I'm glad so many have come here today. I know everybody has a choice about what they can be watching this afternoon. So hopefully this will be a better use of your time than being sucked into the day's news. So I appreciate Dagmar's request that people just go ahead and raise a hand. I want to add to that by saying, please feel free to unmute yourself and just go right ahead at any time. I absolutely will not mind being interrupted. I very much want to try to get over the barriers that Zoom gives us to be as interactive as possible.
And let me add, Aaron, just as we said, right, you will have some breaks in between where you stop screen share if we don't need a slide. So maybe in between have everybody on the main screen and then of course return if possible.
Yep, I will try to do that. There are a few natural breakpoints and I'll make sure to pause at those points, but that shouldn't preclude you from diving in if you want to go deeper or if something was unclear. Awesome. Okay. So I decided to give this whole kind of talk a philosophical wrapper, and I invite you to embrace a challenge with me that I have in mind. And that challenge is here in the title of the talk. Are there things that we can learn about the brain that change our understanding of behavior? Or is behavior perfectly fine without us? By which I mean the attempt to observe, study, and model and explain the regularities that we see in human motor control. So, does a neuroscientist have anything to add to that picture? People do interesting things, and I want to show you some of the things that I find most intriguing and that I hope the work that my colleagues and I have done can speak to. We deliberate. I like to think about the activity of playing a game like chess, where the sensory motor aspects of the game, the ones that you can observe by taking measurements of what somebody actually does, do not tell you the whole story. There's rich internal life going on. There's deliberation, planning ahead, social cognition, thinking about your opponent, their skill level and their stance that particular day. All that comes through in a very simple motor gesture. Okay. Another interesting thing that we do is quite the opposite end of the spectrum, where the motor behavior is about as rich as it could be. It is skillful, it is flexible, it is, artistically elegant, and it results from years and years of training that must be sustained through continual practice. And if you ask an expert performer what they were doing while moving us to tears, they might say, well, I was thinking about my grocery list. Okay, so, another aspect of human behavior that fascinates me is our capacity to improvise, to kind of be locked into a sensory motor milieu and to spontaneously allow new things to emerge, whether that's speech or painting or whatever it may be. These are things that I would love to understand how the brain can do. I'm fascinated by them intrinsically, but I also care very much about their neural basis. Okay. And why do I care about why the brain does these things? Well, one reason is because I'm just curious. I find reductive mindsets to be intrinsically satisfying. Okay. Second, by understanding the brain, I believe and I hope, although there are fewer examples of it than I wish we had, but I believe that by understanding the brain, we can design better therapies for stroke recovery, for development. In the case of brain-computer interfaces, if we want to engineer a neural prosthesis, maybe by understanding how the brain does it, we can do better than if we only relied on behavior. And then, ultimately, I wonder, as we have some examples through the history of science, when we understand the reductive phenomenon, does that influence how we think of the original phenomenon itself? So maybe, just maybe, by understanding the neural basis of motor control, we can change our understanding of motor control. Okay, so I'm slowing down to create room for you guys to interrupt. I reject the entire premise, or gee, that's not interesting. I'm going to turn on CNN instead. This is a good time to interrupt if you want. Otherwise, I'm going to keep on rolling. My standards for explanation come down to the brain's currency, in my opinion, which is the action potential. Okay, this is a well-studied phenomenon that I hope you're all familiar with, whereby neurons emit a voltage deflection, which they use to communicate. Action potentials drive synaptic plasticity. Action potentials drive memories. Yes, memories through synaptic plasticity. Action potentials drive motor control. Eventually, they cause the muscles to contract once passed through the spinal cord to the junction with the muscles. So action potentials are the basis. That's, for me, that's a sufficient reduction of cognition and motor control if we can get to that level. Of course, many people try to go deeper to molecular and genetic bases, and I celebrate those efforts, but this alone would be good enough for me. The methods that I use to try to get there with my colleagues, monkeys performing brain-computer interface tasks, and neural population analyses, which has been a true watershed, a paradigm shift in this field, looking at the activity of lots of neurons working together, which opens up a whole world of geometrical intuitions. Linear algebra, dynamical systems, statistics, machine learning. We can bring all those tools to bear at long last to try to understand neural computation. Here's a monkey and an example of some of the things that these very capable animals can do. And even though our research is relevant for BCI control, the animals are not impaired or paralyzed or anything like that in the studies that I'll describe. They're highly trained, they work every day, and they work with us for 12 or 14 years while we collect lots and lots of neurophysiological data, which we do with a multi-electrode array, this 10 by 10 grid that you can fit several of on the head of on a penny. And you can see an intraoperative photograph where that device is implanted into primary motor cortex. The type of data that we get from this implant is shown here, where time is running along the horizontal axis, and each row corresponds to the timing of an action potential from one of the 100 electrodes we record from. Those are our raw data. This is a good time for me to stop and find out if anybody has any questions. I'm going to abstract from this level of action potentials pretty quickly, so I want to make sure that everybody knows what our raw signal looks like.
Have you told us what the task is yet?
Nope, we're going to get to that pretty soon. The tasks are all brain-computer interface tasks.
And here you suggest you have 96 channels and each of them is plotted. So you're using all of the channels?
Yep. We turn off a few for technical reasons, but in everything I'll show, it's about 90 that end up being useful. There's just noise, some for reasons we don't understand, some have cross-contamination or, no signal on them.
Your arrays in primary motor cortex?
Yep, for everything I'll describe here, this is primary motor cortex. We also do premotor cortex, but those are different stories from the subset we'll talk about today. All right, everyone's on board?
Out of the 96 channels, how many are usable? Some of them are not going to be near anything, right?
We take them all. The ones with less signal on them don't do any harm. So of the 96, 20 or so are absolutely fantastic. you can hold the same individual neuron for months just by good fortune. Another 30 or 40 have clearly task-related signals on them. And then a dozen or so don't do much, but we keep them around and they don't seem to throw off our statistics any.
And how long can you leave these implants? How long do they last?
Yeah, we get 2 to 4 years from them. In humans, thankfully, they last longer. The same devices, I'll show you in the next slide, the same device is also used in clinical BCI trials. And in humans, they last 5 or 6 years. It's really wonderful. They need to last 50 years, but so far they're giving us 5 or 6. Brain-computer interface, as someone alluded to, is the experimental paradigm that ties into everything I'll describe today. I'd say about half of the lab is doing BCIs. That's what we started with first. So those stories are more developed and ready for prime time. The other half of the lab does straight up arm movement control. And those stories are not in here tonight, but once Dagmar and I do our best, those stories will be part of the storyline as well, along with Salah and Rashida and Reza and others working with us. So for everything I'll describe today, the animals are doing something quite unusual and rather interesting. They're not making arm movements at all. And yet, I think I'm going to tell you something about how the brain controls movements. So wish me luck with that. So we're recording in primary motor cortex while animals are sitting with their arms comfortably at their side, thinking about moving a cursor on a computer screen. Here's an example of a paralyzed human who is doing BCI control. So she has a motor neuropathy. She has 2 electrode arrays implanted in her motor cortex. And by thinking about it and learning through practice, visual feedback, she's able to steer this very expensive robotic limb to move the piece of chocolate into her mouth. The monkeys don't have a robotic limb. I worry what might happen, but you can see this is fantastic enough that she can feed herself a bar of chocolate and be utterly delighted by that after years and years of being motorically helpless.
So 2, I wasn't aware of that she has 2 implants, both of them in motor cortex.
So I don't know. I think one is, what I think is going on is that she has 4 implants. Each is a dual-headed array, so 64 electrodes on each. And I believe the setup is 2 in motor cortex and 2 in somatosensory cortex.
Okay.
But I know that when they're doing BCI control, they're using the M1 electrodes primarily.
Okay.
Yep. And the reason I knew once, but I have her and the newer participant, Nathan, shown here, confused. I know that Nathan has 2 M1 and 2 S1, and I just don't remember if they did that with Jan or not. So she has 256 electrodes through 2 connectors, and it may be 2 different brain areas. Okay, this is a wonderful technology, but we're not ready for clinical. This is nowhere near a cochlear implant or a deep brain stimulator in terms of the pace of technology. Other questions? Did someone have?
Reza.
Yes, I have this question. I don't have much sense how large the motor cortex is. These electrode arrays, do they cover like hand regions or arm regions, or they span multiple,
Yep.
I don't know, segment regions?
Great question. Yep, great question. In a human, they barely cover the hand region. That's my understanding. In a monkey, because we're interested in the proximal component of arm movements, just because it's easier to study, they cover shoulder, elbow, probably a little bit of finger, but hopefully not very much trunk. And 1 multi-electrode array is enough to give us basically the entire contralateral upper limb in a monkey. And in a human, one electrode array is only going to give you most of the hand area. And you can see that obviously the goal here is grasp control, right? And Andy Schwartz, who's one of the people heavily involved in this work, has been working on that, toward that for many, many years, both natural control of grasp and also BCI control. And Neville is here. Neville and Andy are pushing forward together in that direction. So that's a long answer to your short question. In the data that I'll be showing you, it's all upper arm, shoulder, elbow, where the electrodes naturally reside. Okay? Anyone else, feel free to dive in. I'm about to start the science that I want you to appreciate today.
But
So
you could control this artificial arm with electrodes in any area of the brain. Do you get better control with it in the motor area?
what a great question. So after we finish chatting today, I will get back to work on a grant whose premise is to explore whether other areas can provide good BCI control. Wouldn't it be fun to do BCI control from prefrontal cortex? Wouldn't it be fun to do BCI control from visual areas? What are you doing when you try to control a BCI from a visual area? So this is a question that is it's a very subtle question, and it's a hot question in our field. There's some evidence that rodents doing simpler tasks can do BCI control using V1, visual cortex. So let's see if monkeys can.
Yeah, just to sort of follow up on that same question, what I've heard about these BCI things is that we put them in motor cortex, and so there's this intuitive sense. It's like, oh, it's in motor cortex. It's controlling a hand. You're tapping into your brain's hand controller. But there's a big, powerful machine learner on reading the outputs from those neurons, and there's a decoder machine, neural network, and that decodes the neurons, and then there's another one that controls the movements. And what I have heard is that we can humans are just very adaptable, and you can put EEG net, and I can control a cursor with that too. And so what I have heard again is that the putting it in the motor cortex doesn't really. I mean, maybe people might learn faster. I guess that's the assumption, but like it could just be that it's just like a machine learning sort of thing, and we're adaptable enough that we can figure out how to, over time, we can make the mapping work that the thing does what we want it to do.
Thank you. These are the issues that are at stake. So one possibility is the brain is pretty equipotential, and all you need to do is have someone smart enough, structure the feedback appropriately, and just sit back and wait, and they'll figure it out. The other possibility is that the apparatus for dexterity is refined in motor cortex. Not all cortex looks the same if you look under a microscope. Maybe there's something in the details of that circuitry that makes it really particularly good for sensory motor learning. But the first study that I want to show you questions the premise that anything is learnable. So let's go into it. And I think, John, your question is going to be one that's well worth returning to in about 5 or 10 minutes. Okay, awesome. So
there was another question that actually disappeared. Is that taken care of? Mohammed was the person.
Thanks for mentioning that. I can keep it until the end, but maybe I ask it now. So how was the elbow and shoulder controlled then in that robot?
Sorry, ask it again. How is the elbow and shoulder controlled in the video I showed you?
In the robot, yes. So I guess you said that the array was controlling the hand, not elbow and shoulder.
Yep. So between your intuition about it, Mohammed, and John's intuition is exactly the space we want to explore. So if I can paraphrase Mohammed, he's saying, well, if you're in finger motor cortex, obviously you can't control the elbow and shoulder because that's not biomimetic. And John is saying, and I think Joel is suggesting perhaps, you can put these electrodes anywhere because people can learn. So in between those is exactly the space that we want to try to explore. More technically, what they did was to calibrate the BCI in a manner not too different from what I'm about to show you, and relate neural activity to behavior through a decode algorithm. And what you tend to find is that if you allow the algorithm to do its magic, just about any neuron can contribute to just about any joint angle. It's these are, you have the 256 dimensional space of neural activity that's mapping into the 10-dimensional space of robotic control. And how you fit that model is buried in the details. But let's keep these issues in mind. I want to talk about how the brain changes when you learn a BCI. And I want to say that the way that we would study learning is by first finding a skill that requires the brain to change. Second, seeing how the brain changes before and after you've learned. And then third, showing that those changes are actually relevant. So I'll try to do that in the coming slides. My colleagues and students, it's nice to see their faces here. We've got Byron Yu and Steve Chase. I've worked with Byron for almost 20 years now. We trained together in Krishna Shenoy's lab as postdocs. Steve Chase, who trained with Andy Schwartz and is now independent, just got tenure, thank goodness, at CMU. No question, but still a relief. Emily Obi, who's a research faculty member at Pitt, and postdoced with me and Steve and Byron before having her independent position. Patrick Sattler, Kristen Quick, Jay Hennig, Darby, and Matt, all graduate students. Some of them have moved on, and Jay is about to finish and actually come up to Harvard. So you'll get to know Jay, I hope. Okay, great. So let's go into an example, the setup for how we use a BCI to study learning. An animal is trying to move a cursor on a computer screen. And to do so, he's generating patterns of neural activity that he hopes are appropriate to move the cursor in the direction and at the speed that he desires. It's our job to design a good BCI decoder to allow him to do that. So I'm going to show you how we evoke learning. It's geometrical. So I'm going to pretend we only have 3 neurons, and I'm going to pretend the animal is only doing 1D control, just so I can put it on the postage stamp in front of you. So we think of this as a mapping from 3-dimensional space down to 1-dimensional space. We record neural activity in a sliding time window of 50 milliseconds. We count the number of action potentials that occur on each of our channels during that 50 millisecond window. And we render that as a point in a neural activity space. It's actually 90 dimensions for the 90 electrodes, but I'm calling it 3 dimensions just so you can look at it. I've got to make my little annotations go away. All right, so lots of windows through an entire day or an entire training session give us lots of points in neural activity space. And this type of geometrical way of thinking about neural population activity is going to be crucial. So if it's not clear, please just make sure you ask. Okay.
So if you're counting spikes within a 50 millisecond window, doesn't that kind of, I mean, how many spikes can you get within a 50 millisecond window?
Gee, 20 or so.
So then whatever you get out here is going to be it's going to be digitized and like from between like integers between 0 and 20.
Yep. And then, as a technicality, I don't think it'll matter for any of the conclusions that I'll try to make today, but all of these data are z-scored so that the firing rate range of individual neurons is not a contributor to any of the conclusions that we'll draw. So you can think of each neuron as exploring its dynamic range centered on 0.
And sorry, just one more. In the 90 that you're using, these are all channels that hear spikes. Because my understanding again is that some of these Utah arrays, the messy ones, they don't get spikes necessarily.
Yes. So we have the good fortune of all the experiments that I'll describe were done fairly early in the lifetime of the array, when we could rely on, if not individual neurons, small clusters of neurons. We could have taken local field potential, filtered it, and taken the power and the high gamma frequency, and probably seen the same results. LFP is pretty good for BCI control, and LFP signals are probably not too different from aggregate action potentials.
But then that
If
would
we
be
do the opposite,
going against your philosophical framework that assumes that spikes are the currency.
well, so I like spikes, and I'll take them all day long. If you give me LFP, I'll grumble, and I'll do my best with it.
Fair enough.
I spent too much time around Christoph Koch as a graduate student to totally discount LFP, but my brief foray into LFP was very difficult and not particularly rewarding. So I take spikes whenever I can get them. Cool. Okay, so I hope this
use non-overlapping windows for your estimate per 50 non-overlapping windows. So the resolution is 50 milliseconds of these data.
Yep. I have a great colleague at USC, Mariam Sanetchi, who has tightened down the window to 1 millisecond and gotten some really fast, nice BCI control. But you have to do a lot of, you have to be a real signal jock to be able to make use of those signals. They get noisy when you get down to that level of granularity. And it's probably not the brain's noise, it's probably just our measurement noise. But 50 milliseconds is a nice trade-off between pretty fast cursor control without a lot of jitter. So that's what we settled into, and we've never gone back to consider that assumption. Okay, so here's what we do. First, we have to calibrate our BCI, and we do it by moving the cursor ourselves on the screen. All the animal has to do is watch. He probably thinks he's in control, but he's not actually in control. He's just watching the cursor while it moves. And in an effect that's not too different from mirror neuron cells or action observation neurons, if you've heard of these, neurons in motor cortex also seem to fire while the animal is watching the cursor drift, after the animal already understands that he can move his arm to move the cursor. So we look at this wonderful paper by Paul Chisak and John Kalaska as giving us a neural basis upon which this effect rests. So when the animal sees the cursor move to the right, you get patterns of neural activity. I just arbitrarily made one large so you could see it. Patterns of neural activity that are in one region of neural population state space. When the animal watches the cursor move to the left, there are patterns of neural activity in a different region of neural population state space. So building a BCI decoder is a simple matter of fitting a line to that relationship. So now what this means is that whenever a neuron in the region that's near the right-hand edge of this line, whenever a neuron over here fires, it's going to move the cursor fast to the right. Whenever a neuron over here fires, it's going to move the cursor fast to the left. Okay, that's our setup. And that allows the animal to move the cursor really well. He's happy, we're happy, but then we induce learning with a simple manipulation, which is to rotate that relationship. Okay, so now the line that represents cursor velocity has changed its orientation in neural population state space, and that forces the animal to do something different if he's going to restore good cursor control. These patterns that used to move the cursor to the right now project onto this line in a way that moves it to the left. And if he wants to move the cursor to the right, he has to generate a pattern of neural activity that used to do nothing. I hope the manipulation is clear. If it isn't, let me know.
Is it cool if I ask a quick question, Dr. Batista? What type of human learning, if you apply it to normal sort of motor control-based learning, like error-based learning or Hebbian learning, what type of learning do you think this is most close to?
Yes, all of them. Sorry, that's a cute answer. But here's my belief about human motor learning. It is all of them at the same time. It's error-based, it's reward-based, it's prior history, it's you know, whatever motif of learning, it's what's the word for it? Coming from exogenous learning. Dagmar, I can't believe I'm blanking on the name for this. This cognitive form of learning when you aim to one side or another, knowing full well that you're trying to defeat the manipulation that you've been given. Explicit, implicit, error-based, cerebellar dependent, basal ganglia dependent. Yeah. They're all going on at the same time, and extremely clever behaviorists have designed paradigms that they declare isolate them.
Okay,
so
I guess that, but why would you think that this, this relatively sort of artificial BCI sort of rotation, why is that representative of sort of human motor learning at all? I guess is the second half of that question.
I don't know that it is. I can't tell you that it is.
Okay.
Yep.
That's great.
But what I can tell you is whatever learning related changes happen here have to be in primary motor cortex. They can be throughout the whole brain, but we've created this little bottleneck. The changes that the brain is going through must pass through these 100 neurons because there's no other way for them to influence behavior.
So in a sense, you're showing that the same neurons adapt and learn new behavior and potentially can switch, meaning in the same substrate, there is neural substrate, there is more than one behavior possible and then simultaneously possible.
Absolutely. Yep. I actually have some of the newer stuff that I'll get into in a few slides asks exactly that question. How can the same population of neurons support different behaviors? Again, couched as control of different BCI mappings. And we'll definitely get into that. Yep. So David, this is a cool trick that we can play. And when I'm feeling philosophical, which I am wont to do, as Dagmar pointed out, we have swapped out a difficult problem for an easy problem that we hope is relevant. But you know, we're no different from somebody who takes a slice of brain tissue and puts it in a petri dish and hope that this is also true in the, you know, vibrant human brain.
Excuse me, Aaron.
Yep.
I'm not sure if you said this or not, but is the animal receiving any reward?
Yes, they get water for doing this task. Their first water of the day comes from working for it. And then if they haven't had enough, we come back an hour later and give them the rest. So they're motivated to do this behavior by regulating their water intake. Okay. Right. Although sometimes we think that they find aspects of this intrinsically motivating. And, you know, when the puzzle is at just the right level of difficulty, it seems they're quite willing to keep going all day on it. There's just something, I mean, we all know that there's something naturally rewarding about motor learning. And I think that's happening here also. But we play it safe by making them earn their first water of the day by working for it. Okay, so it did not escape your notice that there is structure to this distribution of points that I'm showing. And in the true 90-dimensional environment where, I'm just showing you a 3-dimensional schematic for it, that low-dimensional structure is 10-dimensional. We call it the intrinsic manifold. Lots of people have recorded from populations of neurons and applied dimensionality reduction, factor analysis, or principal components analysis, and found structure like this. Okay. So one question we ask is whether that structure is telling us anything real about neural computation. And let me show what I mean by that. So we created 2 different types of BCI perturbation. One that respected the low-dimensional structure that we observed in the population, and the other type violated the low-dimensional structure that we observed in the neural population. So now the question is, if we challenge our monkeys to learn a new BCI mapping, a rotation of the original mapping, do they learn it faster if it honors the low-dimensional structure that we observed at the start of the experiment, or does it not matter at all? All right, so here's some videos from my lab. This is a monkey doing one of these BCI tasks. This is with the decoder calibrated to work as well as possible. And they're doing pretty good. They pick up the targets at about 2 seconds each. Here is what happens after a perturbation has been introduced. The animal's ability to move the target fast and straight, the cursor straight to the target, has been impaired, okay, which creates a gap in performance, which can drive learning. It's both error-based and reward-based learning for sure. Okay, so here's what happens after about an hour of practice with a perturbation that respects the underlying structure. So that to move the cursor straight to the target again, all the animal has to do is explore within his predetermined intrinsic manifold. And here's what happens after about an hour of using a BCI mapping that required the animal to form new patterns of neural activity that were outside of his intrinsic manifold. He's not doing it. He's doing it much more slowly, and sometimes he doesn't get there before the trial ends and he has to start again. So by looking at the low-dimensional structure that's present in the activity of a population of neurons, this thing we're calling the intrinsic manifold that is found through PCA or factor analysis, we can provide new BCI decoders that are readily learnable if the animal only has to explore within that space, and not learnable if the animal is required to create new patterns of neural activity that would take his neural activity out of that low-dimensional space. There's also, incidentally, there's a nice after effect, which you can see for the within manifold perturbation, which to me looks like a signature of error-based adaptation style learning, but that's a tenuous connection at best. For the outside manifold perturbations, where we saw relatively little behavioral manifestations of learning, although we have reason to believe they're trying to learn, they snap right back. They seem happy to get the intuitive mapping back so their performance can continue.
Aaron, we have a hand. James.
Yeah.
Thank you. So I have a question. I'm just trying to wrap my head around what the principal components mean in this case. And I'm wondering, so you talked about, you had z-scored the data. Does that prevent, so I'm wondering if the space that you found is, it in some way means that these are the neurons that can actually fire and have signal. If some of the neurons don't have signal, is there a way that prevents the alternative space from being just firing rates that can't be generated by neurons that don't have signal?
It's a great question. So when we pick these mappings, we choose them very carefully to make sure that the demands we're placing on the neurons are reasonable. And there are lots of factors that we constrain the search by. One of them, you're absolutely right, is to make sure that no neuron is asked to fire outside of its pre-existing dynamic range. Okay, and there are other constraints. We make sure that the decrement in performance is not so large that the animal can't even get started. Like if we made it flat out orthogonal to the intrinsic manifold, he wouldn't even get any signal, so he wouldn't be able to learn. So we dial in all these parameters. It still leaves us with something like 10 million possible mappings, and we just pick one and run with it for the day. Reza, yeah.
I'm just curious, was there any kind of physical movement accompanied these BCI movement? Was the monkey, I don't know, doing like that to move the cursor that way? Or
Yep.
I don't know, something physical?
There is almost no movement. They're twitchy little guys. They like to squirm in their chairs, so they're always doing something. But the hand movements, we have markers on the hand, little optical tracking markers on the hands. Hand movements are minimal, and they don't seem to be particularly goal-directed. So we didn't, for example, observe the animal trying to, move his body to steer the cursor. And we've actually had lots of debates about whether that would help or hurt them learn how to do this stuff. So to the best of our knowledge, they're not thinking about this in terms of what muscles do I have to contract, which I think gets to the issue that we were talking about earlier. Why use M1 at all? Other questions? I hope that this result is clear. We summarize it by saying that when you try to learn, what you can learn is somehow influenced by what your brain can already do. So it's not just musculoskeletal limitations on what you can learn, it's also neural. And it might actually be a pretty simple rule. You look at the low dimensional activity, and that constrains what you're able to learn in a short period of time. Mohammed, yes.
That intrinsic manifold that you showed, which I think goes back to the idea of a neural state space.
Mm-hmm.
And then you mentioned something about PCA. So is that showing the PCs? So you're not looking at the other component that the PC is giving you, so it gives you like two temporal or spatial components. So which one is the one that you're depicting in that figure?
Yep, it is. If you think about PCA, it is exactly that. Okay, factor analysis is a slightly different model, but any PCA intuitions are going to apply here. And for the first few factors, PCA and factor analysis are finding the same dimensions. So what you're looking at is distributions of neural activity that have been fit with a low dimensional structure. And we simply we take dimensions of neural activity until we have enough to account for about 90% of the shared variance. And it turns out that number is about 10 dimensions. So we ignore time to build these plots. Even though these points are in fact connected over time, we treat them individually. Other people include time in their dimensionality reduction, and very interesting insights emerge when you do that.
I think Alexi was first, and then Nicole and Joel.
Hello. I wanted to wonder, are there any ways to generalize the notion of the neural state space over several monkeys? Or what is the notion of this state space and similarities across monkeys?
Yep. 10 or 20 years ago, I would have said, well, that's that's science fiction. But now I think that there's something very, very real there. So we are intrigued by the fact that the dimensionality is always about 10. That's across animals and it's across tasks. We think that those dimensions might be pulling out something stable from animal to animal. It might be, if we're comfortable with the idea of a synergy, it could be that these dimensions have identified synergies. It could be that these dimensions have identified axes of volitional drive. It could be, as I'll show you, that the dimensions are capturing something about inner states, like anticipating the reward or your level of overall arousal. So those things, which I can put words on, will, stably transfer from one animal to another. So I believe that you could say, I've recorded the neurons in one animal's brain are clearly different from the neurons in another animal's brain, but they'll have enough similarities that I could potentially align those spaces between animals. And Chethan Pandaranath, who's a friend and colleague of mine working at Georgia Tech, believes that he can build a low dimensional dynamical model from one animal and fit another animal's neural data with it pretty well, which means you could potentially calibrate a decoder on one animal and it would work off the shelf for another animal. So this thing I thought was science fiction has some basis to it.
Yeah.
Nicole?
My
Joel.
guess was, okay.
No, Alexi, go ahead. I'm sorry. It's just hard to tell who's talking. Yeah.
Just to comment, my
yeah.
guess was whether it is possible in some future science fiction to say that there are tasks, motor tasks, that humans, for example, cannot learn, and which would be more or less generalizable for all the humans, or
yes,
the majority.
Yes. Thank you. Maybe that's a better answer to David. I've built you a model for things people can't learn. And I did it by looking at their brains. There are plenty of things that people really have a difficult time learning. You can describe to them what the goal is, and you can be like, what's your problem? I told you what to do, and you still can't do it. You know what I mean? So maybe we're tapping into something that's like that. Dagmar, are you keeping track of?
I am. Yeah. So Nicole was next, Joel, and then Mariam.
Okay. That's what I thought.
You kind of answered my question when you talked about how one of your colleagues was thinking of creating a decoder on one animal and then using it in a BCI on another animal. So it seems like these, the constraints are pretty consistent across at least monkey populations with regards to our learning.
Yeah, we have found that M1 is stably about 8 to 12 dimensions in all 4 animals where we've looked so far. When we do dimensionality reduction on the human M1 data, the dimensionality is roughly the same. Maybe it's 15 or 20, but it's in finger motor cortex where you might expect more independent degrees of freedom. Yep, so the particular manifestation will of course differ from one to another, just like the pattern of pixels on all of your monitors are different, even though it's all my head, but you can find the same underlying structure. And as long as you can align those structures, you can transfer from one to another.
Okay, Joel? Where did he go?
He's muted. Joel, unmute, please.
Would you predict if the animals were paralyzed, they would not reduce their performance? And secondly, how stable are these? Is this learning? If they were idle for a month, do they come back right where they left off, or is relearning required?
It's been amazing to me how little relearning is required. They're very difficult experiments to do. You have to cross your fingers and hope that the neurons on the array don't go away. But when we've had an opportunity to look 2 months later, getting a little ahead of the story, I'll say it now, the outside manifold perturbations, which take weeks to learn, are stable insofar as we've been able to look a month or 2 later. The within manifold perturbations, which we think are probably more like adaptation, are kind of relearned every time. There's relatively little savings or retention if you return to them. And then sorry, just repeat the first part of your question for me.
If the animals were paralyzed, does that impair their performance at all?
Yeah, we wonder about that a great deal. Not enough to actually do the experiment, but for us, it's a very interesting thought experiment. This is only visual feedback, and you may be aware that as part of the effort to build a dexterous external arm, people very much want to apply artificial sensory feedback to replace missing proprioception and haptics. The question is, would that help or hinder learning a purely abstract task like this? And it's like a Necker cube. I can convince myself that one view is right, and then the other view immediately snaps into focus. So I'd love to know.
Miriam.
I'm trying to understand what it means to move on this manifold. Basically, you've done the manipulation of moving on the manifold or orthogonal to it. But imagine like you have this manifold with bumps and valleys, and you can actually move through the bumps and valleys without respecting the manifold. I think effectively that would translate to dropping out dimensions from that 10-dimensional space. So have you tried with fewer number of dimensions? Is the direction is what matters, or really you have to respect the shape of the manifold?
Yep. So these are just absolutely wonderful questions. And this is exactly the type of fun geometrical thinking that can yield, I think, new insight. You know, not only can we play fun games by giving animals mappings that respect the topography, we can also, you know, ask what it is that the brain is doing that would give rise to structure like this. So I'll just give two very brief answers to kind of go in the direction that your intuitions are taking. The first one is to ask whether there are pockets, hotspots, and dead zones within the manifold. So the picture that I've given you so far is a cloud that we fit with a hyperplane, but not all regions of that cloud are explored equally often, okay? There's structure that relates to the 8 targets, but there might be structure that's intrinsic to the circuitry of the brain. We're just beginning to think about that and form hypotheses and do causal tests with that. Okay, the other, you know, you talked about this idea of tunneling through a landscape. I'm not going to show it today. I have slides at the end if anyone wants to see them, but when we add time into our dimensionality reduction and challenge animals to go against the temporal sequencing of these patterns, they can't do it. Yep. So they can't.
Dynamical.
Not only is this a collection of clouds, it's also got flow lines on it, and you can't reverse those flow lines. All right. If I appreciate that nod. That's actually very gratifying to see. I'll try to put up some slides at the end to you know, give you a look at what that looks like.
Thank you.
So there's 2 more hands, but I know that Aaron has a whole lot more new things. So after Maddie and Carlos, we'll let Aaron go ahead again.
Okay. Mine should be quick. You kind of like alluded to it right now. My question was going to be, since the results are showing that the monkeys can't just learn anything, the way I'm trying to think about it is as if all the neurons in the brain aren't just connected to each other. There's this, for one neuron to be activated, something else needs to have happened before it. So is this sort of like equivalent to a not fully connected neural network, but like a convolutional neural network? Is that how you think about it? Or do you think there's another reason why the monkeys can't learn it?
Yep. I think that is probably exactly right, that we have shown, lo and behold, that the brain is not densely connected. Yep. So I think that these, you know, what you really want to know is the matrix of synaptic connectivity, but we can't get that. In 100 years, they'll have it. What we can do now is infer that we are dipping into a network, and we can ask questions about the general properties that that network has.
Could I ask one more question then? Sorry.
Yeah, please.
So when you do PCA, you also get the weights of all these neurons. So you know, you understand which neurons have higher weights, so they sort of fire together more often, kind of thing. Could that not give us an idea of that? Okay, if neuron 3 is firing, then neuron 4 is also probably going to fire. So maybe that could be the connectivity.
Yes, I like that idea that you can. So the loadings, you know, each dimension here. Let me try to draw this. So is it Manny, is pointing out that this factor, this, vertex of the hypercube is nothing other than a linear weighted combination of the neurons that make it up. So it must be that those neurons are more likely to be synaptically connected to each other. And I think it'd be the problem is the neural recordings are not going to uniquely determine a network. There are going to be lots of network architectures that are identical with that would give rise to the observations that we made. So figuring out that ill-posed problem is a computational person's dream and an experimentalist's nightmare. But if you want to try, give me a call. Yeah, but your intuitions are exactly right. We should be able to infer something about the network architecture from the particular dimensionality that we get. I should say that Brent Dwaron, working with Matt Smith and Byron Yu, is doing exactly that, applying factor analysis to artificial networks with known configurations. It's great work. There's some papers out and more to come. Carlos, Rashida, yes.
Well, thanks for sparking all this wonderful discussion. I wanted to ask, when we talk about the intrinsic manifold, that's the intrinsic manifold with respect to this particular reaching task, right? So in theory, if the monkey were doing other behaviors that are not part of this task, those might be seen as a sort of off manifold, right?
Yep.
Okay, so so I guess, yeah, I just wanted to ask you, do you think of these as sort of like task-specific manifolds, and then is it sort of learning within that task-specific manifold? If you had another task, do you think, yeah, that we'd have another manifold and another set of on and off manifold perturbations?
Yes. So Carlos, I'm nodding and saying yes because you're asking a great question. But I am willing to stake the claim almost as a matter of faith that this 10-dimensional structure is robust across everything the animal does all day long. The problem is I would need $100,000 or to convince one of my friends to allow me to use his apparatus to record from the animals 24/7 in their cages. I'm willing to bet the manifold doesn't change because I'm a true believer. But I think a lot of people would put down money against me on that bet. Okay, what we do know is when they're sitting in their chairs doing a whole variety of different tasks, let me back up. We always see this structure. If you allow them to change the posture of their arm, okay, they're still doing BCI control, but all they've done is to put the arm in a different posture. That's another dimension. Okay, so that's evidence that might get me to abandon my faith, but I stick to it. I say, okay, so it won't be 10 dimensions, it'll be 11 dimensions. Okay, fine, 4 limbs, 14 dimensions, but that's it, no more than 14. Right, but this is an experiment I very much want to do. I just don't own the equipment.
Well, that's a really interesting prediction. It'd be really interesting to see how that pans out.
Yep.
Can I just interject here? This long-term retention has always been one of my interests because, right, we all know that we don't forget how to bike or to ski. And so, in fact, I think these are just any example will do. And we have a very simple experiments where we showed amazing retention over 8 years. So the question always that has troubled me, is there some correlate of kinematic features, right, that are the signature of a person in a specific skill that is stored somewhere, somehow in the brain? And the behavioral evidence is just in our face everywhere.
Yep.
But can we conceive of this in neural terms? We should, but how?
Yep, that is a spot on way to put the question that is really etched in my mind. Where in the brain, you can think of, what's the term, the engram. Where is the engram for riding a bike? or dancing, or you know, you name it, motor skills that we do again and again that we never lose, that never get overwritten. Are they in motor cortex? Are they the details of the synaptic circuitry in motor cortex? Are they, you know, spinal cord, cerebellum, basal ganglia, all of the above? Yeah, I mean, we got to find the motor engram.
But you might just use your monkey and at the very beginning, and then whatever, as long as the encoder works 4 years later, and subject the monkey to another 15 studies, and then just test the study that you did 4 years ago again. And if that manifold arises and it's different from what the monkey did in all those intervening years, then that would be a first experimental demonstration.
Yep, that's a really wonderful suggestion, because that is so simple to do. Those data you can get for free in just a week or two. Yeah, there will be some technical issues, but we know how to solve them without re-estimating the same manifold. But I'm glad this is being recorded, because many of these suggestions have been really thought-provoking and wonderful, and I don't have to try to remember them while continuing to engage with your ideas. But that's, yeah, you'll remind me of this when we meet in the next couple of days, Dagmar, because I want to make sure that I suggest that to Emily.
So you have more to show. I'm so eager. Rashida has lowered her hand. Can we leave Rashida's question for later? And
Rashida,
actually,
you email it to
sorry.
me?
Professor Vargas Irwin scooped my question, so I lowered my hand. If the dimensionality is actually task specific.
Yep.
Like if they were following like a cursor along a sphere, would it increase? I was wondering. But.
So if what I would love to do is I think you were asking a slightly different question from the one that Carlos was asking. I think you're asking, can I change the task that the animal is doing in the chair and change the dimensionality of the population? That is a quite different question and rather cool. The challenge is going to be to find a task that's significantly more difficult, but the animal can still do it in the chair to get a affirmative answer to that. Yep. But I do acknowledge right away that this is a very simple behavior. It's just moving a cursor fast and straight to one target. There's a lot more that we could do without me needing to buy all that fancy equipment I was mentioning.
And
cool.
then
Almost question, Aaron, shoot ahead.
Rashida, you will email me, please, and we'll chat next time we can. Good.
Definitely.
I can't tell you how delighted I am by all of these questions, because this is the point in the talk when I've laid out all of the conceptual framework and y'all got it. And you're thinking in exactly the ways that our minds have gone over the last 5 or so years. And it's just extremely gratifying, in addition to being some really, affirming some really wonderful directions we could go in. Okay, but for today, in the last 15 minutes, I'll stick around as long as you guys want, but I think Dagmar is going to start letting people go pretty soon. I'm going to give you some choices about where we go from here, okay? So I can't do them all. I'm excited about all of them. So I'll ask the questions, and then maybe we can, I don't know if Zoom lets us do a survey or if people can put up hands. The thing in blue is looking at the outside manifold perturbations and asking whether they can be learned if you simply give the animal enough time. The answer is they can. And when you learn them, you do so in a very interesting way. You generate new patterns of neural activity that are appropriate for moving the cursor. So that's the summary, and I can show you the evidence if you want, but it's published. And then the last two are things that, well, C just got published. Just got accepted, so it'll appear soon. It's on bioRxiv now. B is the following, and then I'll get back to C. Dagmar asked a question. A lot of you are probably wondering, what if we ask the animal to learn several different mappings? Do they jointly consolidate? Do they interfere with each other? So we can look at that. I'd be quite excited to show you those slides. They're pretty quick. And then C is going to be, thankfully, it's on bioRxiv now. Thankfully, will appear soon. And this is a serendipitous realization that there are internal state signals present in our motor cortex recordings. Not what we expected to find, but it kind of hit us between the eyes and we needed to track it down. So A, B, or C? How are we going to do this, Dagmar?
I have no idea. I have no idea.
Okay, who wants to look at the data on long-term BCI learning? And maybe Dagmar, you can do just kind of a quick
I'm
count.
seeing in the chat, okay, I see
great.
a bunch of Bs. And
All right, we're doing B.
We're doing B.
And then maybe we'll come back to the others.
Oh,
Okay.
Carlos wanted C. Well, Carlos, wait. B first.
You might get C. So I encourage you to go look at Emily Obi's wonderful paper from proceedings 2 years ago. And she did the heroic thing. I'll just show you the video because we're here anyway. She did the heroic thing of getting the animals to do outside manifold perturbations. So here's day 1. You're just going to watch the same target over 8 days. This begins to speak to Dagmar's long-term issue, although this is 8 days and she wants 8 years, but you know, give us time. So here's day 1. This target is impaired. Oh, gives up. Here he is after 5 days of practice and some coaching. It's getting better. So these things are learnable. Here's day 14, and he's pretty zippy. These things are learnable, even though learning them requires making new correlations among the neurons we record from. All right, moving on to C. How do multiple skills jointly consolidate? So this is work that is led by Darby Losi using data that Patrick and Kristen collected, only the within manifold perturbations. So Darby's question is this: if you learn something new, does it change how you consolidated your old memory? Okay. I don't think we know. There are enough neurons that you could presumably encode something new without touching the old stuff. Or maybe there's some type of interaction between memories, which maybe it's sometimes helpful and maybe sometimes it's deleterious. So let's see if BCIs give us a window into that. So here's Darby's cute way of asking the question. This is from his talk at Cosine last year. What if you're a good skier and then you decide to hop on a snowboard for the first time? You give up on that. Maybe you learn, maybe you don't. You know, after a couple of days, you say, no, thanks. I'll just stick with skiing. Are the neural activity patterns that control your ability to ski influenced or not by having taken a few days to learn how to snowboard? Okay. So to think about this in our geometrical neural terms, you can imagine that in neural state space exists the substrate for task A. Along comes task B. You form a new, whatever you want it to be, a new configuration of neural activity or a new attractor, however you like to think about it. Your neurons have exhibited patterns of activity that are appropriate to performing task B. We saw that in the earlier work. Now, when you go back to task A, where do you land? Okay. We actually already know the answer to this thanks to Emilio Bizzi. It's wonderful to see you, Emilio. Back in 2001, he showed that individual neurons change their tuning. If you study the tuning, the preferred direction of premotor and motor cortex, I'm in motor. I'm pretty sure Emilio was mostly in premotor. And then you go do something else, force field adaptation or BCI learning, and then you look at the tuning again, it's changed. So now the question is why, okay? And does looking at the neural population help us answer the question of why? So here are 3 possibilities. Maybe, so the claim is, we know from earlier work that the way you express task A2, the second rendition of task A, is different from how your neurons express task A1, the first rendition of task A. Why? Maybe this is just a random change. It's all in the null space. It doesn't matter for behavior. Or maybe task A2 bears a signature of having learned task B. Okay, some type of joint reconsolidation. Or maybe, you know, you're just trying to return to task A1, but you get lost on the way and time runs out and the experiment is over. So these are the three hypotheses that Darby concocted. And here is his geometrical interpretation for that question. Okay. Because it's BCI, we can, I mean, you can do these things conceptually. And Robert Agemian has a nice conceptual framework developed with Emilio for thinking about this for real arm movements. For BCI, the concept is the real deal because neural activity patterns map directly to behavior. So for task A1, for task A, whether you're in block A1 or block A2, neural activity patterns projected onto this red line determine the cursor's velocity. And of course, you'll realize that there's an equipotent null space where neural activity patterns along that vertical line I drew have the same manifestation on behavior. Okay. When you switch to learning task B, your goal is to hit the equipotent line for task B. And you can see for this little cartoon schematic how the neural activity patterns would have to evolve over time to give you that performance ability. Okay. So now we can think about different hypotheses in terms of where task A2 activity lands relative to task A1. Maybe the task A2 activity ends up back where it started or partly there. Maybe it ends up just somewhere else. Or maybe it ends up in a very particular position, which continues to give you good performance through task B, should you ever encounter it. I think it's a logical question, and a BCI gives us a tool to ask it, because the BCI lets you do something very special, which is you can see whether neural activity is suitable for a behavior you're not actually performing. And it's really hard to do that with just natural arm movements. So what do I mean by that? So here's the animal doing his BCI task. He starts off with neural activity suitable for map A because we calibrate the decoder. This is the intuitive mapping that he knows perfectly well how to use. He learns to get better at doing map B. Then we give him map A again. There's that little after effect while he readapts or figures out what's going on and his neural activity becomes suitable for map A again. So here's the BCI trick. We can project the same neural activity through the other decoder and see how performance would do. So we can ask during block A2, is neural activity more suitable for map B than it was during block A1? And the answer is that it is. Okay. So this is a schematic, this is real data, but this is just a graphical illustration of the idea. If you take the map A activity and project it through map B, the cursor trajectories are just kind of wherever. But if you take the map A2 activity and project it through map B, they're also pointing toward the target. They're pointing toward map B's target. Okay. So it seems that there is some evidence that you've reconsolidated map A's knowledge in a way that has a persistence of memory from task B. Okay. This is work in progress, so that's not a lot of detail, but I hope that gives you a sense for how things are looking so far. I forget if I included
what
the
was
population.
the actual two tasks that were examined?
Yes, map B is a within manifold perturbation. I should have drawn a little schematic. So this is rotating the BCI plane, the readout plane, so that it conforms to the intrinsic manifold, but still requires some learning.
Yeah, because from experience, right, some of these tasks, A, B, A, Some of these have positive, others negative transfer. So the question could be, if you did a similar but in a principled way different B task, whether you could explore under what conditions learning B is helpful or is detrimental. What are the limits of that plus, well, I don't know, reorganization?
Yep. It is the direction I would love to go. So this is all a post hoc analysis, meaning you don't have the tight control that you'd love to have in an experiment. But this is all within manifold perturbations, and Emily has been looking to see if Darby's rules are also true for outside manifold perturbations. And that's the context where the question you're asking is more tractable, because there's a much bigger range of manipulations. So we started off with very simple geometrical rules, like if the projections, like if the angle between the two mappings lies, acute, maybe there's transfer between them. And if the mappings are greater than 90 degrees apart, obtuse, then it may be that the animal has to switch and he can't find a consolidated solution. That simple prediction did not bear out. Okay, this is one of those things where you're like, well, are there technical problems, or is that really just not the right way to think about it? So Emily is forging on to see if she can find something there.
Okay. And maybe as I just caused the break here, we are nominally closing in on 6:30, which remains to be our target time, but we invariably flow over. So I just, I would like to hear more, Aaron, but I do understand if some people just want to, well, quit. So I would suggest we continue, knowing that we're greatly overrunning time and many people may just need to leave.
Anybody who needs to leave, you got dibs on questions. And if you leave, I will certainly not be offended. And I'm very, very grateful that you came and listened and contributed your ideas. I no surprise. And as usual, I've enjoyed this immensely, even over Zoom. And I'll leave you with my little philosophical nugget. And then if you want, I can go back and talk a little bit about one of these other stories. So, with the caveat that this is all BCI control and not what you might really want, which is, real florid, natural motor behavior full of dexterity, we've been able to use this Petri dish to see some interesting things. We've seen how the brain changes when you learn. Okay, we've seen how it respects the underlying constraints that exist prior to the beginning of the learning, and violating those constraints takes you a lot longer. Some evidence that reconsolidation may actually be different from the original consolidation. We have that already with individual neuron tuning curves, and those principles are also applying at the population level, with the new insight that the reconsolidation seems to preserve your old abilities, help you with the thing that you recently learned. Okay, so I don't know, I'm sure I haven't changed your impression of how motor skills work behaviorally, but I do hope that work like this, looking at the neural basis for motor learning, might actually get us eventually to think about what it means to have a skill behaviorally. Okay, so who's got questions? Oh, right, and then just a shameless plug for the work that Dagmar and I are embarking on with Mohsen and Salah and Reza and Rashida and folks on my side, Raid and Pat Lachlan. Our hope is that we can gain insight using these population tools into real arm movements. And maybe, just maybe, some of the BCI observations we'll observe, but will still pertain. But that's, of course, an open question. Okay, so I will do the third part, part C, for people who might be interested. But those of you who have to leave, go ahead and ask questions, or just ring off and I'll say goodbye. Mohsen has a hand up to either say goodbye or ask a question. And then Emilio did also.
No, I have a question, but apparently the background doesn't show. Oh, this is my hand. So it was up all the whole time, but it's not visible. So I think Dagmar sort of asked my question partially, but I want to specifically ask about the phenomenon of anterograde interference, where people learn sensoromotor transformation, which they call it task A, for example, a force field to the left, and then it's followed by task B, which is the force field to the right. It's the opposite direction, and they just can't learn it. And so I was wondering that, can you see that in these manifolds, that what happens, because these are sort of, I think these are sort of within manifold perturbations still. It's just you're changing the direction of the perturbation to the exact opposite. And what happens in this neural dynamics that you just can't learn that?
Yeah, that's a really cool question. So the picture that's coming to mind for me, and I have no evidence for this, but consistent with the things we've seen, think of a cloud of neural activity that has only a limited ability to reshape itself within a day, within the time scale at which adaptation works. But what you can do is move that cloud around. So Matt Golub calls this reassociation. You've taken the same spanning space of neural activity patterns and you've optimized them for a new task here, learning one of our WMPs. So imagine that applies in one of your anterograde interference paradigms. You might take that cloud of neural activity and go make it really suitable for one task, which puts you at jeopardy, makes even more difficult to learn something that would have been equally available to you had you done it first. It's easy for me to imagine that we can concoct a hypothesis like that, like there's this fairly non-deformable thing that all you can do is shift it around or move around within it.
Yeah, I think, well, the interesting thing is that if people associate each task with a different context, and these contexts are, people have tried so many different contexts, but for different contexts, if you associate each task with one, you are able to actually learn two opposing perturbations. And the question is, and I know you probably don't have data for this, but I just want to know what you think about this, that is the context just a different input to the same population of neurons that changes everything? Or is it just, it sort of allocates a totally different resource for the new task? It's just a different bunch of neurons that are allocated for the opposing task that you're able to learn both at the same time.
Yep, this is a very exciting picture. So think of context as allowing you to stack up memories. So my wonderful mentor, Krishna Shenoy, has done a VMR adaptation task with the arm in two different postures. And using population recordings, he's shown, this is a bioRxiv paper, he's shown that when you're in one posture, your neural clouds deform in a way that's appropriate for VMR learning. When you go to another posture, the shift in neural population space is orthogonal to the plane in which you learned originally. And it can support a non-interfering VMR learning, so that by changing the arm's posture, you're kind of clicking into one or the other of these readout modes. So it's almost as if you can think of the arm, these other dimensions, these output null dimensions, as indexing and stacking up different sensory motor mappings. So Sulu Sun is the first author on this. It's a bioRxiv paper now, and that's indexing by the arm's posture. But just like your intuitions are, Mohsen, you could imagine indexing by the context. Oh, I'm in the place where I snowboard, so let me click in this set of memories. And now I'm in the place where I rollerblade, so let me click in this set of sensory motor mappings. And these can all coexist, and maybe they're indexed in a relatively simple way. Yeah. So you're asking about a very interesting thing.
Thank you.
Tyler.
Hi. I had a question about the nature of the perturbations and the learning that sort of corrects for them. I guess if these were humans, maybe we could ask, but as a monkey, then you can't. So I don't know if you have another way of identifying whether the learning would be like, let's say they knew to go this for up and this for down, and then you perturbed it so that like the same neural activity that would have, that when they do this makes the cursor go up. Is that like a conscious decision? Like, is the learning like, oh, I know that I can go like this to make the cursor go up? Or is there actual neural connections that are changing such that consciously they imagine the same thing, but the neural activity sort of in a hidden way like reconnects to now get the target under this new condition?
Yep. There's a long history of training animals to make an anti-reach or an anti-saccade where they'll see a stimulus on the right and they have to move to the left. It's really hard to get monkeys to do that. Many people have tried, and thankfully they've succeeded because it's a really wonderful way to do a sensory motor disassociation. For humans, you tell them the rule and they pick it up pretty quickly. I believe that for humans, 180 degrees is easy to do just because you cognitively understand, you use kind of explicit information. But I think 170 degrees is really hard to do.
Yeah.
So, I think David made the point that there are different learning mechanisms at play. And I, for the sake of argument, I swatted it aside and I said, they're all active at the same time. But humans definitely have explicit learning, and I'll bet monkeys don't have explicit learning, or at least it's really impoverished.
So if you do this perturbation, you are guessing that the monkey is not just realizing, oh, I need to think up to go right or something.
Yeah. And for example, that wouldn't generalize. If I taught them up means down, they wouldn't then say down means up. That would be another couple of months of training to get them to understand that.
Yeah. One other part of that question, just to follow up on Tyler's question, like, do you believe that there's there's actual changes in synaptic connections happening that's underlying this type of BCI learning, or is it just facilitation of existing connections in different neural patterns?
Yep.
I mean, is there any evidence that point us in either direction?
I'm a synaptic plasticity skeptic. I think that, maybe just because I like to be contrary, I think that there's too many times we equate learning with plasticity, and there's virtually no evidence for that. I think that there's ongoing synaptic resculpting in the brain, when we sleep, through homeostatic mechanisms, teasing out the changes in synaptic weights that really are learning and memory dependent from all the other stuff that's happening is very, very difficult. And flipping it around, these networks are vast, like the number of computational states that you can achieve in principle in motor cortex, even within a 10-dimensional subspace. There's an enormous number of different states that can all be reached just by driving the area differently. So, of course, I would love to say that our long-term BCI perturbations engender new synaptic plasticity, but they certainly don't need to. You could hit those new states just by driving the same network in different ways and exploring new modes within it. It's hard work. These days, it can only be done in rodents to actually see synaptic changes, but I think people are way too quick to say, he learned, therefore there was plasticity. So I'm certainly not making any claim like that.
Miriam has a question.
Miriam, yeah.
So I'm trying to, again, understand the two spaces that you're actually moving in the previous part of the talk and this part of the talk. So now it looks like you're actually you have another state space that you're moving in. So should we think, is this the same space and these are packets in the same space, or should we should you think about this as some sort of a hierarchical, basically two sets of state spaces, like hierarchically kind of laid out? I'm not the
Yep, we have a strong desire to record in other parts of the brain while animals do these BCI learning paradigms. We want to record in premotor cortex, and we also want to record in prefrontal cortex. And that's because we're inspired by a hierarchical view of sensory motor coordination and learning.
What I actually mean is you kind of, in the first part of the talk, you showed that there is this manifold, and then you can move, it's better to respect the manifold and move it in
Yep.
on that manifold. In the second part, you have these I said you have these subset of manifolds, like one is for task 1 and the other one is for task B and the other one is for task A2. So each of these are themselves, if you get the center of these subsets, basically, you have, you're moving basically on another manifold, right?
Yep.
So
yes.
That's what I mean by hierarchy. I don't mean the hierarchy in
Yes.
the brain sense, basically. Yeah.
Yeah, I gotcha. All the learning tasks are two-dimensional BCI mappings. And for this study, the within manifold perturbations, those two-dimensional mappings are embedded within 10-dimensional space.
Mm-hmm.
And to go just a little more technical, because we've z-scored the data, they're all touching at the point where baseline neural activity is 0 cursor velocity. So they're two-dimensional planes that must intersect in a line because they intersect in a point. Okay, cool. And then for OMPs, they are two-dimensional planes embedded in a 90-dimensional space. So the hierarchy is, you have this outermost space, which is the observed neural activity, the possible neural activity given our recordings. And then you have the actually observed neural activity, which is 10 dimensions. And then, each day's experiment is some two-dimensional plane that falls at least somewhere within that 90-dimensional space. But, there's also another hierarchy, which is the rest of the brain, right? And it's still a two-dimensional plane embedded in the 10 billion space of the rest of the brain. Yep. And then when you think about the learning task, it's an exploration in a 10 trillion dimensional space, if you think that you're in the space of synaptic weights. Yeah, so all the, yeah, I like how much you value these geometrical intuitions. This is where the hypothesis generation happens.
Thank you.
So
Okay,
Aaron, with
yes.
all the questions, can you give us a very breezy overview of that last number C? Because otherwise Carlos will be very angry with us. But
Yep.
I certainly understand that it's getting long. And maybe let me just call for an applause. I should have done that before. So let's just applaud our speaker.
Thank you. That's, as I said, it's you bring together wonderful people, Dagmar, and it's really great to have been able to interact with everybody. Go if you want. I promise to do Jay Hennig's work justice in about 5 minutes, and I'm not going anywhere. So if you got questions about any of it or Jay's work, it's we will stick around until Dagmar cuts us off. Okay, so
sorry. I
yeah.
do have a question, and I do have to go pretty soon. So this is all very cool, and BCIs in general are very cool, but I remain relatively unconvinced that this has all that much to do with motor control. It's definitely something they're learning something, but it feels more to me like it's some kind of puzzle solving that's happening, and I would love to be convinced that this is telling us very much about motor control, and not just that if you have 100 billion dimensional neural space, that it's possible to, with the right decoder and the right feedback, to just find some way to fluctuate that in a way that correlates with this very low dimensional task.
Mm-hmm. John, thank you. I have no idea what this relates to.
Okay, good.
So just to be totally clear about it, we recorded from M1 and we created an artificial mapping from M1's activity to a cursor movement. It's not totally artificial because we could figure out how to make it so the animal could learn to use it in a jiffy without any learning. And then we messed with that mapping, making it even more artificial. And then we observe something that resembles learning. It comes on gradually and it has an after effect.
Mm-hmm.
And then we found that if we take the thing that the animal couldn't learn within a day, he does learn it if you give him 2 weeks. I have no idea what we did.
Yeah.
Okay. So I do know a few things. Not all BCI mappings are equally learnable. Okay, we see that in M1. I'm certain it's true if you do EEG learning. People are very clever individuals. Jen Collinger has allowed us to try some of this with her human participant. He doesn't learn OMPs faster than the monkeys do. So it's not like you can just reach in cognitively and solve the problem in a jiffy.
Mm-hmm.
Okay. I think that M1 is a very special brain area. I think it is both low dimensional and especially accessible to volitional control. I don't think it's going to be as easy to do this in prefrontal cortex or visual cortex, but I'm willing to try with the taxpayers' money if the NIH will let me. You know, what would it mean to learn using V4? Is that you know, having visual hallucinations? What would it mean to learn? Maybe PFC learns everything as long as you structure the feedback properly. Maybe
there's
PFC
just
doesn't have a
structure.
low-dimensional substrate.
I mean, like, you know, some aspects there's some conscious control happening somewhere in there, and maybe it's just that there's, you know, we learn how to fluctuate the larger scale statistics in a way that maps onto this task in some way. And I'm very interested to see about if you were to drop those Utah arrays into other parts of the brain, if there is something, because, you know, if thinking of the parts of our brain that sort of map onto conscious control, the motor cortex seems to be a pretty good candidate. But it has the, you know, like I said, it's very interesting, and you're definitely looking at something, but I think we have a similar sort of curiosity of like what it actually is. It's hard to know.
Yes.
I've done experiments. I've been subject to experiments that do things like you do these motor remapping things where they do some weird mathematical remapping of high dimensional like hand posture and body posture onto some sort of things on the screen, and the sensation is puzzle solving more than, you know, motor control. So it's, again, interesting, but one of those sort of things where I leave with more questions.
And thank you for sharing many of them with me. And, you know, I reach out if you want to keep the conversation going. I spent, when we were doing this work and trying to, you know, get it out there and publish it, I was defiant, and I had my stack of arguments for why this was motor learning and why people like Dagmar should care. And then I got tenure and I stopped caring about making the argument that it's like motor learning. It's something else, potentially something else entirely. I think your instincts are right, that it might be more like puzzle solving. And I don't know if anyone's going to care about this work in a few decades, but maybe it's not people who are trying to understand motor control. You know, you can imagine, who knows where the technology may get us, especially if we can do high resolution, non-invasive electrical or muscle recordings and use those as interfaces. Maybe motor learning is just, you know, where it started, but not where it goes. I don't know. But remember my setup. I'm looking at internal deliberation during chess playing. I'm looking at improvisation. These are puzzle solving scenarios. You know, skill learning. It's the most tenuous link might turn out to be motor skills, but I hope we find out in the coming years.
Yeah, I while you were talking under intuition, I Googled it, and rhesus monkeys are tool users. primitive
Yeah.
ones, but they are tool users. And I wonder if that could matter, sort of the ability to, learn the external dynamics of some other thing might be what's going on here. And I wonder if you were to give it to like marmosets or something like that, if they would have any hope of solving a problem like that.
I'm pretty sure they wouldn't. I've spent some time with marmosets, and I'm
They're
pretty
pretty
sure.
dumb.
Yeah, and they're very they're good at what they do, but what they do is very specific and narrow, and yeah, I'm not terribly interested in the neural basis of shrieking loudly. So no marmosets for me. Sorry, I mean, rhesus monkeys are very sophisticated creatures, like you're talking about. There's another interesting aspect of what you pointed out, and I'm going to use it to leverage into part C. So those of you who can stick around, please do so. Which is M1 is sitting there. I mean, John mentioned how well positioned M1 is, kind of anatomically and functionally. It's receiving input from all over the brain, and it has one function, which is to control the skeletomotor system. Okay. That's what M1's doing. There's projections to the viscera. There's presumably some eye movement and vocal control has to go through M1, but mostly it's there for the arms. Okay, and even walking, a lot of locomotion is left to the spinal cord. In order to do that, M1 needs to get a lot of information from all over the brain. So it's perhaps not too surprising that Jay found enormous signals of internal states just sitting there in these M1 recordings that Patrick had made, 5 years earlier. And I'm very proud of the fact that, primate neurophysiology data sets have, been able to enable discoveries long past anything we had in mind when we collected them. It's beautiful data to explore, and lots of serendipity comes out of them. So look at this somewhat confusing plot, and let me walk through it. This is a low-dimensional visualization. It's about to turn into a movie. And what you can see circled here in green is the distribution of neural activity patterns when the animal was moving to one target. Maybe it was to the right, I don't actually know. Okay, while he's using the intuitive mapping. So you can think of, I'm not showing you the BCI decoder, but you can imagine the BCI decoder might have been kind of stacked up over here so that all of these points moved it in the appropriate direction. But there's just some natural variability to the neural activity patterns. Along comes the within manifold perturbation, and the neural activity jumps. Before any learning has happened, the neural activity just jumps. We thought of it as like a surprise signal or an arousal signal, but those terms were already taken. So we ended up calling it an engagement signal. Reviewers said it's not arousal because it doesn't fit this definition. We said, okay, fine, it's this other thing called engagement. But if you look target by target based on color, there's the natural variability while the animal is doing the doing BCI control with the intrinsic manifold, which suddenly jumps when the decoder is switched. And then Jay made a great animation of this, so you can see this relationship. The 8 targets are well distributed in neural activity space, corresponding to what they need to be to hit the 8 targets. And then there's an organized jump whenever the manifold changes, whenever the BCI mapping changes. Okay, for reasons I'll describe, this was described as the neural engagement axis. And there's an aggregate axis, but also for each target, there's an individual axis. So here's what it looks like, and it covers the span of natural activity patterns during intuitive mapping control. So if you take each of the 8 targets and you project their neural activity onto this target-specific engagement axis, you can see that across all targets, the following interesting thing happens. Right when you introduce the new BCI mapping, all the neural activity jumps and then gradually settles back along that axis. And while that's going on, learning is happening. It wasn't just when you switched the BCI mapping That's where Jay spotted it first. But it's also if Emily or Patrick paused the experiment to go in and make a change, this jump occurred during all of those epochs as well. Okay. Happens right at the start of the experiment, happens relative to pauses, happens when the new BCI mapping occurs. So it's not a learning related signal, it's something else. Oh, and then Jay got some old data from Steve Chase's lab and found that that signal was in there also. At the beginning of the experiment, or when Steve and his students switched to a visual motor rotation with hand control, neural activity jumps. Okay. And this engagement tracks pupil diameter really well. So M1 is sitting there receiving a signal about an internal state that seems to relate to surprising events in the environment that aren't necessarily related to learning the new task. We weren't expecting to see this, but it kind of makes sense that it's there, because if you're going to control movements, you have to know the overall state of the brain. One quick point I want to make about this is some of these random, they're not random, they're very well-structured jumps. They're just random relative to the BCI mapping that we've challenged the animal to learn. Some of these jumps happen to push you in the right direction. They push you toward the solution to the BCI you're trying to learn. Other jumps push you in the wrong direction, and they're counteracting anything that you might need to do to learn. Okay. And we can actually see by looking at the orientation between the engagement jump and the target the animal has to try to learn, that target's position in neural activity space, you can partition them into helpful jumps in blue and hurtful jumps in red. And those give you different learning curves, different times to saturation. And this effect is longer, outlasts the actual engagement signal in the first place. So that's Jay's story. We're glad after years of his hard work digging into this effect that it's finally going to show up pretty soon. It's on bioRxiv now. And I think the moral of the story is that there are internal state signals present in M1. They're easy to detect because we have a BCI. So we know their orientation relative to what we've challenged the animal to learn. Sometimes they help and sometimes they hurt, even though they're not directly related to the learning. Which brings up, I think, a really cool prospect, which is if you give someone a BCI for motor function, maybe you can find some off-label uses like, you know, changing your level of engagement or arousal or making yourself happy. Who knows? All right, so that's part C. Now I'm really going to stop. I'm so grateful to all of you for sticking it out to the bitter end.
It was not a bitter end. You stopped with something that can make us happy. So I think
there you go. Right, exactly.
So why don't you turn your screen off and just show us without the slides, and maybe a concluding comment or still a burning question, but I think dinner is calling. Or as I said, a glass of wine, going out to a nice restaurant after a long and engaging talk. So that would be so nice.
Anybody who wants to keep the conversation going, just drop me a line. I'd be happy to chat more about these things. Thank you, Dagmar, and thank you again, everybody. Really appreciate it. Thanks. You guys
Thank
are
you,
awesome.
Aaron. Thank you, everybody. And thank you for your all amazing endurance hanging in there. So it's great. And just to say that again, I think for me at least, it works better than expected, these virtual action clubs. So thank you. Thank you for being there and being lively and interesting. That
Yep.
was for everybody, including the speaker, of course.
That's exactly my feeling about it. Thank you all very much, and take care.